Everybody's ready now? Let's go. Okay. The very interesting thing is that one part of, I should say, of my system for, I should say, gene expression, it gives me a vector with 0, I should say, identified that. It seems that I don't know what's the reason. I do not get any error message, but it doesn't pick it up for, I should say, for the gene expression profiles. It's okay. With the next step, I can catch up with you. But if you have that, I should say, the gene expression profiles, I just gonna just work, let's say, for example, go to this line. You can see the result. I cannot show you the result, but if you have the gene expression profile, it should work fine. And after, we just go to write the first machine learning algorithm, which I can join you there at that step. Okay, it's going to be that step. It's going to be somehow an unsupervised machine learning, which by doing so, you are going to have a very interesting heat map to visualize the distances. Always remember, the measure of the distance is more important than the choice of the machine learning algorithm. For example, if you go to write a code like that, where was I? Here. If we just run a code like that, I call that Euclidean distance, EUCD. I use a function dist, which expresses the function which I created. You do not see that, you do not have the same function, huh? Do you have the same function or no? That's fine. Okay, whatever you have there for the distance, when you click in there, you have the Euclidean distance. You will see that it shows up at the global environment. Then the problem is that when you just want to do an unsupervised machine learning to visualize, that's a very important steps. By doing, I should say, unsupervised learning, you find the genes which they cluster together, they stand close to each other, then you find the correlation between the gene which causes the cancer with the genes which you have no idea, and it gives you a clue to do further analysis. That step is very important. That's why I told you the distance is more crucial, because it gives you some insights to some new genes which causes the cancer. In order to do that, you have to input as a matrix. Then that's what the second line does. It converts your data sets into a matrix. That's why I call that UEM-M. It's a matrix. You can see the dimension of your matrix depending on cutoff rate. My matrix is 86 to 86. Because my cutoff rate was extremely high, 99%. It means that I almost killed all the things which is my data sets. Make the things computationally faster. And now, I just want to just do some painting. That's the artistic part of the machine learning. You choose a palette because you are just going to visualize, you're just going to pick up some colors. Now you are the Da Vinci of, I should say, machine learning. That's what you have to install the package. R Color Brewer. The package is being installed. And now I just go to the library command. Everyone has a code, huh? Yeah? You run it, and now you have the palette installed. And now I call the function HMCol. Stands for heat map color. I just use color ramp palette. I just choose red and blue. Because the painting which I wanted to make, I just only choose two colors, red and blue. And my design is that if two things shows up as, let's say, red, it means that those two things are so closely related to each other. That's what the distance means. And if they are blue, it means that they are far apart. It means that they have nothing to do with each other. We do not mean a physical distance. It means a kind of a relation. But the problem is that with this one, the palette in R is designed in a way that the higher intensity, they showed it by blue, the less intensity, they show it by red. We want it to be different. We want higher intensity to be red. And that's what I do. I just go to the HMCol, use a function as ref, HMCol. That's it only reverses Rev stands for reverse. Reverses the design of your palette. And now if I run it, okay, if I run it, now the red shows a higher intensity and the blue shows less intensity. Okay, and now there we are, to run the first algorithm for unsupervised machine learning, to visualize the distances, to figure out how the genes are related to each other. And that's the heat map function. You can use it as a kind of a template. It always runs like that. The minimal, I should say changes to that thing. I'm just gonna run it. Voila, I have this one. You can just come here to zoom it. Isn't that nice? Do you have everything, you see the same, I should say, the heat map or different? Or maybe you should have a reverse one. There should be more red color there. There it is. If you reverse the color, I'm gonna zoom it, and there it is. That's the heat map, the first unsupervised machine learning. You will see, I should say, some clustering on the top. We do not talk about that because it's a very huge, I should say, concepts for the rich, I should say, cut tree, clustering, and classification. Just look at the heat map. Those areas which they have the more intense red color, it means that that's the area that you, as a researcher, you have to focus there. It's lots of things which it shows you. But the distance which we used, it was Euclidean distance, wasn't that? Huh? And look, what it happens there. I just run, I should say, I should say, I just want to use right now another package, install another package. Let me go there. Okay, and also, I just missed one step. It was the Manhattan distance. That's one of the methods which we use for the distance. We just use the Manhattan distance. We can also change it to the distance of your choice. For example, you can just let me just copy paste it somewhere else. I just want to show you how the distance changes the configuration of your data sets. For the, I should say for the distance, the method, I'm just going to write Euclidean. Control Enter. Euclidean distance. Euclidean matrix. D. And as a matrix, I'm just going to run it. And now I'm just going to come exactly do the same heat map for it. Copy paste the heat map. And I'm just going to use just the name of the function, EUM, and run it. It was EUCM. See that if you run it, you see exactly the same heat map because Euclidean and Manhattan sometimes are exactly the same. But now the more fun part starts with, I should say, with Tau Kendall and Spearman correlation. If you please go over there and just install the BioDis package. You see all the package there, huh? And just run the library. It also asks me for the update. I'm just going to run for the updates. run the library, Control F, and to just run the library. Okay, and now just come over that. Look at that, for the Spearman correlation method. I'm just gonna measure the distance based on the Spearman, which is, it finds not the distance between two genes, it just finds, I should say, the correlation which they have. And that's why in cancer genomics, normally that Spearman or the Ken delta are more relevant. I'm just gonna run the function, convert it into matrix, and just run the heat map. Wow, look at that. Your heat map, it looks much way different from the one which you had before. If you zoom in, you will see a different, I should say, tangent. Everyone see the same thing or different? It's running? Because how much was your cutoff rate? If your cutoff rate it was not 95%, probably the computation, it takes more time for you. That was the reason I just chose 99, because I just wanted to run everything so fast. Okay. And now, I'm just going to show you something very crucial. There is a function which it finds, look at that, it comes after the heat map functions. Because when you just run the heat map, if you have, let's say, the cutoff rate, the cutoff variance which you have, if you, for example, run your algorithm on the whole data sets, looking at the heat map is very confusing. You see thousands of genes are clustered, and you do not know which gene relates to the gene of your interest. There is a function there, this is called closest.top. For example, you just come with an ID of the gene. Later you will see that you can change this ID of the microarray into a symbol of the gene. You have a command later coming up. That's going to be your gene of interest. And you say that it's going to search for, by the way, for any commands, if we just put your cursor before the parenthesis, press F1, it opens the help menu for there. And you see what kind of arguments you have to put inside the function. Just pressing F1 when you put the cursor before the parenthesis. And you see that for the first argument is the X, which essentially is a gene of your interest. The second argument is a distant matrix. Dist.map means a matrix which you created. We created, I should say, the Spearman matrix, which is Spear M. And the top means the number of the genes which in your heat map comes as close as to your gene of interest. For example, if that gene, 39120 underscore AT, you can choose anything else from your heat map. Look at your heat map, pick up something, put it there. When I just put 1, and when I enter it, it shows me that thing, gene number 36821 underscore AT. It means that that gene, based on the distance which you measure as a Spearman, stands as close as to your gene of interest as possible. Means that it has the highest correlation based on the distance which is selected as a Spearman. If you choose, let's say, Euclidean distance or Manhattan, you get a different result. And that's why I told you, the choice of the distance, it changes the protocol of your research. Because you may find a different gene when you choose a different distance, and it may lead you somewhere else. That's why always ask yourself, when you do machine learning in genomics, what distance measure you have to choose. We're just going to go for the last one. It's going to be tau d. I'm just going to control D. That's a tau Kendall. Change it to the matrix. Run the heat map, and you see the heat map is more severe. And now the gene of the interest. You see that I got a gene, and coincidentally for this one, Kendall and Tau, they just got something similar for me because I had very, I should say, a huge cutoff rate. But for you, it should be different. Each distance gives a different genome, which it comes close to the thing which you have measured. You have two more distances, Hamming distances and Kullback linear distances, which is very interesting distances in statistics. I'm not going to cover that, we're running out of the time. Then please, when you get back home, simply control it on those things. I post the slides, you educate yourself of those two distances. They are the best distances in cancer genomics, but we have to just skip them to get to some more fun stuff. We have also a kind of a projection technique, which it shows your gene from that huge dimensions into 3 dimensions. You can use your cursor to move around to see how genes are closely related to each other. Simply when you just got home, just run them. The package for that is mass. You see the package mass and RGL. Run those packages, just run those codes. You see a very fun stuff to play around with. You can visualize the genes in 3D to see how they are closely related to each other. I'm just going to skip that to just in order to just get to the first algorithm for machine learning. Sorry we had to skip. 2 hours for machine learning for cancer genomics is crazy. It requires days, weeks of working there, but that's the kind of taste to see how it works. Okay, we are going to step number, I should say, I'm going to go back to our roadmap. We choose the distance, and now we are just going to select the features. That's what we are going to do. Please, yes.
Question?
Yeah.
How do you choose this? You can try them all and pray?
It depends on your experiment. You have to come with your experiment to see what does it mean if 2 items are similar. Then based on that, it means that, for example, if they show a higher correlation. For example, if your experiment, sometimes you have to use Kullback-Leibler, I should say, information distance, because you just want to see how those distributions are closely related, not the correlation, the distribution of those expressions. Then it depends on your, I should say, experiment. And to be clear with yourself, what does it mean if two genes are similar? Because distance always shows similarity. And wrong choice of distance takes you somewhere which should not be there. That's very crucial. That's always I emphasize. The distance measure is more important than the classifier itself.
So you have to rely on your domain knowledge.
Absolutely. When you just work in finance or bank, always you can use Euclidean distance because that's the handy distance. You don't need to find the correlation. But for cancer genomics, it's a very tough one because you have to have lots of domain knowledge to see what does it mean when 2 genes are closely related. No? Make sense? Super. Okay. Now please scroll down to the section which it says supervised machine learning data splitting. Okay, and now the thing is that that's a kind of a general, I should say, approach. When you have the machine learning, you always, for example, it's like you're having a dinner, you have a plate, you do not eat it all. You split your dinner on your dish, you eat 2/3 of your food, and leave 1/3 for future, for your tomorrow breakfast, for example. Huh? That's exactly like machine learning. You split your data on your plate 2/3. With 2/3 of your data, you train your system. With 1/3 of your data, you try to see the prediction that you made, how efficient is that, how accurate is that, based on the 1/3 of your data. That's a general rule for machine learning. Huh? And that's what we're going to do. We do not follow the approach of 2/3 to 1/3. We're just going to make some samples. The first line of code. Next, if I run it, it shows the items which they have no cancer rearrangement. The second line picks the items with the Philadelphia chromosome rearrangements. The sample 1 makes a random sample of your, I should say, NEG data sets to pick up only 22 items from there. It says sample next, 22 means 22 items from your sample, and replace false. You make your first sample. The second sample, it picks up random 22 item from, I should say, your BCR item from the Philadelphia chromosome. And what do? Now I make my training set. by combining them together using the C command. And that one is there. I created my training set. This is my 2/3 of the food for tonight. 1/3 for the breakfast tomorrow comes here with the test set. But I use a function set difference. 1 to 80 means that 1 to 80, whatever is left, it takes it to the test set of your training set. Now we create a training set and the test set. Now we know what's going to happen next. Everyone is cool now? Everyone's happy so far? You are moving so fast to run the first algorithm of machine learning. One of the things which I have to remind is that for cancer genomics, R is the best package for machine learning. Because some of the features for genomic data comes with the R, which doesn't come with the Python. For example, the package which I have here, maybe you do not have it there. It's ML interfaces. ML interfaces comes only with the R. And you will see that how easy is that to use that package. Everyone has that command there in your, I should say, your workbook? ML interfaces? Everyone has it? Yeah. Okay. Then I'm just going to run it. It means machine learning interface. It's running the package. Any questions so far? As long as it's just installing the updates. Just going to also run the library. Okay, and now look at that. I have to congratulate you so warmly because now you are writing the first algorithm for machine learning, K and N algorithm. How it works? This is the fastest, I should say, training with the machine learning. We do it as quickly as we can. Look at that, how K and N works. K nearest neighbor, that's what it stands for. It means that, for example, look at the graph over there. Look at the star. At the star, that's the thing which you have no idea what is that. You have two different samples. You have the purplish, I should say, circles. You have the yellow ones. And the star in the middle, you have no idea if this is purple or if this is yellow. Which one you think is that? What is your best guess? Is it purple or it is yellow? For our purpose, purple can be Philadelphia chromosome, yellow can be no gene rearrangement. Which one you think? What is your best guess for the star?
Depends on the value of K.
Depends on the value of the K. K there is 3, for example. That was very good. The value of the K. Super, brilliant. But the thing is that if you choose value K equal to 3, means that we have a radius of, let's say, 3 around, I should say, your star. 3 means 3 nearest neighbors. When you have 3, look at that. How many neighbors you find there? Then 3. You search for the 3 closest neighbors. 1, 2, 3, they are the closest in that radius. And you find the probability. You see that the star, when K is 3, it closes, it is close to 2 purple, 1 yellow. You take the votes from it. 2 purple, 1 yellow. Then your classifying algorithm, machine learning, decides, then the star is going to be purple. Because there are more votes from the purple than yellow. It's called a kind of a Bayesian statistics. We do not go through the math, but that's how it is. You take the higher votes of the closest neighbors. It's like you are friends with good friends. The more friends you have, they are good. There is a chance that you are a very personality. That's like that. But if K is 6, what is your guess? I just got that from, I should say, the slide from the Wikipedia. I'm not going to do plagiarism. that's I have to mention. But if your K is 6, as you mentioned, that it depends on the number K, what do you think then in that case? Is a star, is purple or yellow? take the majority votes. Find 6 closest neighbors. Right? 1, 2, 3, 4, 5, 6. Star is yellow or purple? K equal to 6. Who says yellow? Who says purple? That's yellow. Because you see that you have, I should say, 5 yellow and 2 purple. That's the majority vote. And you see, that's a good lesson to take. Depending on the K which you choose, your diagnosis of the star is different. If K was 3, the star was supposed to be purple. If K is 6, your star is supposed to be yellow. Then imagine that you are diagnosing someone with a cancer. If K is 3, you diagnose that person to have cancer tumor, but if you choose K6, then you say that person is completely healthy. Then you see the dilemma here for machine learning. Depending on the K, you have different results. Any question on the K and N? No questions so far? Everyone is happy with the concept? Okay. And now you know the concept, that's time to just put that into some codes to see how it works on the cancer genomics. Okay, we are just going to run the first code of the K and N, and that's the code. I call the algorithm KNN1. KNN1, and you can call it anything. I use the function mlearn. It stands for machine learning. It comes only with the R, not with the Python. And if you do not know what kind of arguments it has to go there, press F1. You see it there. The first argument comes as formula. If you check the formula, the help package there. The formula is that you come for the class. What are you trying to predict is the class. If you remember, the mol.bio was the type of, I should say, the gene rearrangement. Philadelphia chromosome, NEG, ALF4, those kind of stuff. That's what we are predicting. That's a class. That's, for example, in the KNN, it's going to be, let's say, that yellow or purple, I should say, circle. Then you say that the class, I have that sign. I do not know what that is called. That curly, I should say, I know where is that on the laptop. If you know what that's called, just let me know. Tilde, yeah, tilde. Tilde, and I have a dot there. It's very crucial. Dot means that your function y, mol.by is like a y, equals to x. It means that your y, your prediction, mol.by is your prediction. It can be anything when you just do the machine learning. It's a general rule. Your prediction equals to everything. You do not know anything about your futures. You put a dot there and you relieve yourself. Okay, whatever it can be. I do not bother myself to search for the future. Then comma. The next argument is the data. Your data is what you filtered out before. LL filter. And dot method. And dot method there. It's going to be, when you use ML interfaces, the name of the package comes with I at the end. For example, random forest with the machine learning interfaces come with random forest I. K and I, and you choose K. You choose L equal always to 0, but for the K, that's what we meant. K equal to 3 was yellow, purple, K equal to 6 was yellow on that slide. And that's the K we should choose. You can work with it to find the optimal, I should say, precision. We just put it 1, you can change it to 2, 3, or anything. K means the first closest neighbor. If I go back to that slide, it means that if I choose K equal to 1, it means that, for example, for that star, the only closest neighbor, which is going to be this purple one, is taken into the account. K equal to 1. And the fourth argument is train IND. I leave it where it is. I do not just mess around with the arguments, but I only change it with the training set which I created. And now with the training set, I'm training the machine learning to make the prediction. Everything seems okay. I'm just going to control ether. And there it is. My algorithm is run. And I wrote the first machine learning algorithm for KNN, or K nearest neighbor. As easy as that. You can always have a template,